While fungi are important conduits for nutrient uptake, understanding plant nutrient uptake requires understanding root absorption properties. Active transport is the major mechanism by which plants absorb potentially limiting nutrients from the soil solution at the root surface. So here, it's important to understand that nutrient uptake is an active transport process. That means it requires energy to bring nutrients into the plant. As such, nutrient uptake uses energy by the plant. And in part, the reason for this is that nutrients are brought into the plant against a concentration gradient. So if you take a look at concentrations within a plant, they're higher than outside the plant. And to bring nutrients from areas of low concentration to high is against that concentration gradient. And that's going to require energy. Now, plants bring nutrients in and also some leak out. And a lot of the energy that's used is to bring those nutrients back into the plant after they leak out. For example, phosphate leaks out of plants at about 1/3 of the rate that it's brought into the plant. So some nutrients are actually leak out at a higher rate than others. And this can be pretty substantial. If we take a look at nutrient uptake at the cellular level, here's a cross-section of a root. There are root hairs, and the diffusion shell is shown here, for example. Now, there's many different cell layers associated with roots. The epidermis is the outside layer of the cells, and the endodermis is the inside layer of the cells for the cortex itself. Inside of the cortex, you'll find xylem, which are cells, and we've seen those before, that move water, and phloem, which are more important for moving carbohydrates, for example. Now, looking at the cortex a little bit more closely, which is right here, that the endodermis has a waxy layer, and anything for water or nutrients to pass through into the xylem requires that they actually pass through cells. So water has to be actively moved into cells and through the plant. It can't just go in between cells and then into the plant. Same thing as for nutrients. At some point, they actually have to enter into cells before they can be transported into the xylem or the phloem and moved to different parts of the plant. The cortical cell is shown here, and essentially what this diagram shows is that there's transporters that are going to be present in the plasma membrane. And that means for those nutrients to go into the cell, they're going to be brought into the cell via transporters, and that's going to require additional energy. Now, it's important to know that some plant species tap pools of nutrients that are unavailable to other plants. All plants are not necessarily experiencing the same pool of nutrients there, and they don't take up nutrients in the same proportion as other plants. For example, within the soil, there are many forms of nitrogen. So we have amino acids that are present, and these amino acids, there's a number of them. Some of them are low molecular weight, some of them are high. We also have ammonium that's present in soil solution, as well as nitrate. So when we have amino acids, ammonium, and nitrate, all of these potentially can be taken up by plants. But in some ecosystems, you'll find different proportions of each of these in soil solution, and within the same ecosystem, you'll find that some plants will rely on one form more than the other. There are consequences to taking up these different forms of nitrogen. If we think about an amino acid, for example, it has a general formula like that, and that's readily, once it's taken up, incorporated into proteins without much transformation at all. On the other hand, if plants take up ammonium, that doesn't have a carbon skeleton with it, and so then assimilation must occur where that ammonium is then turned into amino acids and linked with a carbon structure. That, though, doesn't require much energy, and much more carbon than would be present to generate the amino acid anyways. But nitrate, on the other hand, is a little bit different. If we look at nitrate, which has a charge of -1, the N in nitrate is +5. In ammonium, we have +1, which means there's one extra proton. NH3 would be neutral as far as charge, which means that this N is at -3 state. That means it's going to require somewhere on the order of 8 electrons to take nitrate and reduce it to the same charge as ammonium. So in that case, if a plant takes up amino acids, there's really not much it has to do to get those put into proteins. If it takes up ammonium, it really needs to just have a carbon structure. But again, the nitrogen itself, not much energy has to be put into that nitrogen to start to form amino acids. Nitrate, on the other hand, requires a large energy expenditure to reduce that to the point where it can be used for amino acids. And so, therefore, plants might have a greater abundance of nitrate in the soil, but would prefer to take up amino acids or ammonium. And so that balance is one that generates a lot of the variation that we see in uptake. So when we think about the costs of taking all these different forms of N-up, that there's costs to reducing nitrate that aren't present in taking up ammonium or amino acids. Now, if energy is in excess, which can happen in a high light environment, for example, there might not be much cost to the plant in reducing nitrate. And there are some species, for example, that will take nitrate up and transport it to leaves. And the leaves are the site of the reduction. So there's lots of light that hits the leaf, and as we know, sometimes leaves, they're going to experience excess light, or light that's in excess of what they can use. And that energy can be used to take nitrate and reduce it to ammonia, for example. So in this case, in some situations, plants are going to reduce nitrate with excess energy, and that's captured within the leaf. And so in that case, those types of plants might actually prefer to have nitrate or be equivalent to taking up ammonia, because once it's transported to the leaf, there's no excess energy cost to reducing it. Plants that are growing in shade, for example, energy is at a premium, and nitrate would be very expensive to reduce. That being said, plants usually absorb whatever they can get. Most plants take up the different forms of N more or less in proportion to how it's supplied. So a plant might prefer to take up ammonium, for example, but if there's lots of nitrate in the system, they'll still take that up and pay the cost of reducing that nitrate. Just like there's different forms of nitrogen that structure ecosystems, there's also different forms of phosphorus, or at least different sources of phosphorus that plants can access. And really the story with phosphorus, as far as uptake is concerned, is that there's different ways to increase the availability of phosphorus in soils. For example, some plants might release phosphatase enzymes, and essentially what that does is it takes organic phosphate and cleaves that, and then releases inorganic phosphate. So in this situation, there might not be a lot of inorganic phosphorus in the system, but there's a lot of organic matter with phosphorus in it, and phosphatase enzymes can be used to increase the availability of phosphorus. Other plants have a different approach, and sometimes they'll use chelates. What chelates do, if you remember, within soils that you can get iron phosphates that are formed, and these precipitate out of solution. Well, so this iron phosphate can still be displaced from the iron, and one way to do that is to release into the soil a chelate, such as citrate. And what citrate does is it displaces phosphate from the iron and puts that into solution. So some plants might rely on using phosphatases if there's a lot of organic phosphorus around. Others can rely on using citrate or malate, something to solubilize phosphorus after it's precipitated out. And in doing so, they can displace phosphate on iron and put that phosphate into solution. There's lots of special adaptations for increasing phosphorus availability. One of these is called protoid roots, and it's called protoid roots because they were first noticed in plants of the genus Protea. But other genera like carex, for example, have something similar, and carex are called dociform roots. But you can see that it's basically the same. You get generation of very fine roots and root hairs that form these dense structures within the soil. And from these surfaces, they secrete a lot of chelates, for example, or acidify the soil solution, and in doing so create localized hotspots of P availability. So this is a place where P availability is relatively high. By forming these dense structures, they can have a localized area of high P availability that's not going to diffuse out such that other plant roots that are there are going to be able to get it. So all you have to do as a plant is take a relatively small volume of soil, secrete a lot of things like citrate into that soil that's going to solubilize phosphorus, and then that phosphorus is not going to travel too far and you'll be likely to get it. So that's one adaptation to increasing P availability, and a specific structure that's used to do that.